In today's lecture, we'll cover amino acids. So this lecture will be introduction to amino acids, and then in the following lecture, we'll be looking at proteins and properties of proteins. Amino acids that are naturally occurring amino acids and constituents of proteins are alpha amino acids. There are 20 naturally occurring amino acids. Actually, there are a few more, but they are very rare. So there are 20 amino acids that are common to proteins found in living organisms. And as I mentioned, those are alpha amino acids. Alpha amino acids, acid means that on carbon, alpha to carbonyl group. That's all nomenclature, alpha, beta, gamma. So if there is functional group, such as carbonyl group, or in this case actually carboxyl group, carbon next to it is alpha carbon, next to alpha carbon is beta carbon, and so on. So in this case, there is amino group on alpha carbon. So these are alpha amino acids. There are other amino acids that are not constituents of proteins, such as gamma and delta amino acids. And of course, a number of other amino acids that have been synthetically generated. Naturally occurring alpha amino acids are chiral. So they are specific enantiomer. And that enantiomer is, as you will see soon, L enantiomer. So when we draw an amino acid in such a way that alpha carbon is part of the chain that points up, then substituent or side chain R projects towards us. So we indicate that with a wedge bond. Alternatively, if we draw this chain as pointing down, so as that alpha carbon pointing down, then substituent R should point away from us, should be represented by a dashed bond. So it's very important to be able to make sure that you represent amino acid in correct configuration, so correct enantiomer. So these are naturally occurring amino acids. Those of opposite configuration, opposite enantiomers, are unnatural amino acids. As I mentioned, amino acids are classified, naturally occurring amino acids, as L amino acids, and that's because they are structurally related to L-glyceraldehyde. All old DL system of nomenclature of designating enantiomers as D or L was related to configuration of glyceraldehyde. Glyceraldehyde exists in two forms that were designated as D and L. In form it's written here, where on vertical bonds we have aldehyde group and primary alcohol at the bottom. Then we have hydroxyl group pointing to the left. In D enantiomer, hydroxyl group points to the right. So corresponding formula for amino acids is one where when we represent amino acid in Fischer projection, same way as glyceraldehyde, where carboxyl group is at the top and group R at the bottom, then amino group, in this case, points to the left. Or in formula on the right of amino acid, in form of Zwitterion. That's a form in which amino acids actually exist, in form of Zwitterions. In form where we have separate charges, positively charged ammonium group and negatively charged carboxylate anion. So in a Zwitterion form, ammonium group is on the left. After amino acids and proteins, we'll cover carbohydrates. And what is interesting is that naturally occurring carbohydrates are D. So while naturally occurring amino acids are L enantiomers, naturally occurring carbohydrates are D enantiomers. And those D enantiomers are structurally related to D-glyceraldehyde. So you can see in case of D-glyceraldehyde, hydroxyl group points to the right. In case of carbohydrates, you can see in Fischer projection, bottom carbon, very bottom one is primary alcohol, CH2OH, and carbon attached to it is one that determines whether carbohydrate is D or L. So if hydroxyl group is on the right, that's D carbohydrate. As I already mentioned, amino acids exist in form of Zwitterion. Zwitterion means that there are charge separations in such molecule. So it's a general term for any molecule that exhibits charge separations, where positive and negative charges are separate. Zwitterion is overall neutral species. And so Zwitterion is neutral form of an amino acid. In Zwitterion, we have ammonium group and carboxylate anion. Each one of those groups is capable of donating or accepting a proton. That means that effectively there are two acidic groups. And that in turn means that there are two pKa values for any amino acid. So besides Zwitterion form, an amino acid could exist in protonated form, and that's form in which carboxyl group is protonated. So loss of that proton from carboxyl group would give Zwitterion. And then third possible form is deprotonated form. Deprotonated form is form in which ammonium group, NH3 plus group, is deprotonated. And here you can see two pKa values for this particular amino acid. This particular acid is alanine, and so those are pKa 1 and pKa 2 values for alanine. First one for deprotonation of protonated carboxyl group, and second one for deprotonation of ammonium group. Now we define isoelectric point as point at which amino acid is electrically neutral. That's actually what isoelectric means. Isoelectric would mean equal charges. We can take that to mean equal charges. So at isoelectric point, Pi amino acid molecule is neutral. And isoelectric point is characteristic for each amino acid. And as you will see later when we cover proteins, also proteins themselves have their own isoelectric points. So Pi, or isoelectric point, is average of the two pKa values for simple amino acids. So pKa1 plus pKa2 divided by 2. Besides ordinary amino acids that are classified, as you will see in a minute, as hydrophobic and hydrophilic, there are acidic and basic amino acids, which have acidic or basic group in a side chain. Such amino acids and some other amino acids actually have 3 pKa constants. In case of an acidic amino acid, Pi is the same as that of ordinary amino acid, which means pKa1 plus pKa2 divided by 2. On the other hand, for basic amino acid, Pi is pKa2 plus pKa3 divided by 2. So average of the other two, pKa2 and pKa3 values. So you should remember that for all amino acids except for basic amino acids, Pi value, isoelectric point, is average of the first two pKa values, pKa1 and pKa2. While for basic amino acids, Pi isoelectric point is average of pKa2 and pKa3. Here is a list of hydrophobic amino acids. Hydrophobic amino acids have nonpolar side chain, and I will describe them in a minute, but nonpolar side chain makes them hydrophobic. That means that side chain doesn't form strong intermolecular interactions with water, and in general we try to basically get away from water. That's very important for properties of proteins. Now, first amino acid listed here is glycine. And there is some debate as to where glycine should be, how it should be classified. Based on its side chain, it's nonpolar. Glycine actually has no side chain. It has hydrogen. Side chain is substituent on alpha carbon that projects towards us. So one on wedge bond on alpha carbon. And you can see here it's hydrogen. So there is really no side chain, but hydrogen itself is nonpolar. On the other hand, lack of any side chain makes the overall species actually rather polar, because it's the two polar groups, carboxylate anion and ammonium cation, that give character to this molecule. So overall it's actually quite polar. And so a lot of authors actually classify glycine as polar or hydrophilic amino acid. So as I said, there is some dispute and there are arguments on both sides. Rest of them, you can see alanine, valine, leucine, isoleucine. Those have hydrocarbon, typical saturated hydrocarbon side chain, and so those are typical hydrophobic amino acids. Methionine is also hydrophobic, side chain is nonpolar, it has sulfur embedded in that side chain. Proline, also hydrocarbon side chain, but you can see it's rather unusual side chain in that it's folded into a ring and attached to nitrogen. So side chain is actually part of the ammonium group. And then we have two amino acids that have aromatic side chains. Those are phenylalanine and tryptophan. Another large group of amino acids are hydrophilic amino acids. Those amino acids have polar side chain, so it's side chain that has some polar group and usually is capable of forming strong intermolecular interactions to molecule of water, typically hydrogen bonding, or at least if not hydrogen bonding, then strong dipole-dipole interactions. And in fact, all of these amino acids form strong hydrogen bonding to water, except for cysteine, which has SH group, that's thiol group or mercaptane group, mercapto group. SH forms strong dipole-dipole interactions with water, does not form hydrogen bonding. While these groups are polar, they are not charged. So they are only polar but not charged. So you can see that serine and threonine are two alcohols, two typical alcohols. Tyrosine is a phenol. So it has hydroxyl group attached to aromatic ring. And then cysteine, as I mentioned, has mercapto or thiol group. And finally, we have two amides, asparagine and glutamine. And finally, there are acidic and basic amino acids. There are two acidic amino acids. Strictly speaking, name aspartic acid refers to protonated form, where carboxyl group is CO2H, protonated carboxyl group of a side chain. This would be aspartate. Under physiological conditions, it's in the form of aspartate. And same goes for glutamic acid. It should be protonated, and this is glutamate. But under physiological conditions, they are deprotonated. So this is aspartic and glutamic acid, or under physiological conditions, aspartate and glutamate, those are acidic ones. Basic ones are lysine, arginine, and histidine. And they all have, as you can see, positively charged group. They differ a little on side chain. Actually, histidine does not. Histidine has imidazole ring. We'll cover those structures in a minute. But you can see that basic amino acids have a little bit diverse structure, and we'll have to look at them in detail in a minute. Now we are going to look at memorization of structural formulas of amino acids, and we start with glycine. And in fact, when we start with glycine, we really want to start with alanine, not glycine. So with glycine, we replace hydrogen of glycine that projects towards us, that's one on the wedge bond, with methyl group. And that gives us formula of alanine. Starting with alanine, we can, by doing appropriate substitutions on that methyl group, derive most of the other amino acids. We will start with hydrophobic amino acids, and if on that methyl group of alanine we replace two hydrogens with other methyl groups, with additional two methyl groups, we end up with what is called isopropyl group, and that's valine. Now, if in the molecule of valine we insert methylene group, CH2 group, between that isopropyl group and alpha carbon, that's molecule of leucine. So that CH2 now that is on the wedge bond, that's the one that has been inserted. And finally, if we move one of the CH3 groups, methyl groups, from the end of the chain to that CH2 group, that's molecule of isoleucine. Iso for isomer of leucine. When we look at other branch, placing phenyl group, benzene ring, on methyl group of alanine, so replacing one of hydrogens of alanine with benzene ring, gives us phenylalanine. And now we are going to make slight digression to polar or hydrophilic amino acids. Placing hydroxyl group in para position or position 4 of benzene ring gives tyrosine. That one is hydrophilic amino acid. So this gives us 6 hydrophobic and 1 hydrophilic amino acid. Now there are 3 hydrophobic amino acids that's the best is just to memorize their structures: methionine, proline, and tryptophan. You could come up with some ways to derive them from alanine, but I think the best is for these 3 just to memorize them. And then we should look at hydrophilic amino acids. Again, we start with alanine. So placing mercapto group on methyl group of alanine, so replacing hydrogen of methyl group of alanine with mercapto group or thiol group, whichever nomenclature you prefer, that's SH group, gives us cysteine. And then the other branch is placing hydroxyl group on methyl group of alanine, so replacing hydrogen with hydroxyl group gives serine, and then replacing one of those remaining 2 hydrogens of CH2 group with methyl group gives threonine. Of course, if you want to examine this in more detail, you can always pause the video and look at it in more detail. And then when we look at acidic amino acids and their derivatives, again we start with alanine and replace one hydrogen with carboxyl group or carboxylate anion. If it's carboxyl group CO2H, that's aspartic acid, carboxylate anion, that's aspartate. And then in aspartate, if we convert carboxylate anion to amide, that's asparagine. So asparagine is derivative of aspartate. Very easy to remember. If you know formula of aspartate, then it's easy to derive asparagine from it. Note that asparagine is one of those amino acids whose 3-letter abbreviation is not first 3 letters of the name. Obviously, that's reserved for aspartate. And then, if we go back to aspartate, if we insert one CH2 group into the chain, if we insert one CH2 group into a side chain, then we end up with glutamate. And of course, conversion of carboxylate group of glutamate into amide group gives glutamine. Glutamine is again one of the polar amino acids. Asparagine and glutamine are polar amino acids, and as you can see, they're derivatives of acidic amino acids. And finally, basic amino acids. Again, we start with alanine, and replacement of one hydrogen of alanine with imidazole. Formula of imidazole is shown below. That gives histidine. Histidine actually has a side chain that has both acidic and basic properties, because one of the nitrogens that has double bond attached to it can accept a proton, so it's basic, while the other nitrogen has proton attached to it and can donate that proton, but also has free electron pair and can accept a proton. So imidazole has side chain that has both basic and acidic properties, even though we classify it as basic. For that reason, it's very important amino acid in enzymes, in active sites of enzymes, as you will see a little bit later in this course. Then next one is arginine. Rather unusual structure. It has 3 carbon side chain, so 3 CH2 groups, and then guanidinium cation attached to it. So basic group is this guanidinium cation. So note rather unusual cation. And finally, there is lysine, which is kind of formula we would expect for typical basic amino acid. Simply it's amino acid that has side chain with additional amino group or under physiological conditions, it's protonated into ammonium cation. So it has 4 carbon side chain and amino group at the end of that chain. So here are pKa values of acidic and basic amino acids. Top 2 are in the top row, 2 amino acids are acidic amino acids, and bottom 3 in the bottom row are basic amino acids. As you can see, they have 3 pKa values, and of course you can stop video here and examine them in more detail. You can see the values for those pKa values, what they exactly are. Now, if you look at different tables, you will find different values. I don't know why different values occur. Maybe different conditions under which they were taken. I don't know. On the test, of course, you will be provided the values that you should use. So you can notice some trends. And you will note that in case of acidic amino acids, carboxylate group of the actual amino acid has lower pKa value compared to carboxylate group of the side chain. And that's because of the inductive effect of the neighboring ammonium group, ammonium substituent. In case of basic amino acids, those values are pretty much all over the place. So you can see that histidine has pKa of 6.0, which is actually pretty low, but that's an indication of its properties. It has both acidic and basic properties. It's what we call amphiprotic species. It's capable of both donating and accepting a proton. In case of arginine, you can see a pKa value of 12.48, so highly basic group, and lysine about what we would expect, 10.53. Value is a bit higher compared for the amino group of the actual alpha amino group of 8.95, and that's because alpha amino group, one with pKa value of 8.95, exhibits negative or experiences negative inductive effect of the neighboring carboxyl group. Now, you may think that these are the only amino acids that have 3 pKa values, but actually that's not the case. Some of the polar amino acids that have groups that could be deprotonated also have their own pKa values. And they are listed here. These are 2 the most common examples, although all of the amino acids that have alcohol functionalities in theory also have 3rd pKa value. So you can see that tyrosine has phenol group, and that phenol group could be deprotonated, so its pKa value is 10.46. And cysteine has thiol group or mercapto group, and its pKa value is 8.37. Okay, and this completes our lecture on introduction into amino acids, and next we'll be looking into structure and properties of proteins.